Okay, good morning and welcome to microbiology. My name is Kristen Green and I will be presenting these lectures. So let's go ahead and get started with chapter 1, which is Introduction to Microbes and Their Building Blocks. So normally in the news, when you are hearing about something relating to microbiology, you're usually hearing about how these microorganisms are hurting us or harming us. You hear about all these different disease outbreaks. You hear about food being recalled due to some type of microbial contamination. We just came through COVID. But what we really don't talk about is how these microorganisms help us. And one of the main ways that these microorganisms help us is by decomposing organic waste. But before I jump ahead and talk about that, let's talk about what a microbe is. So when I'm using the term or talking about the term microbe, I mean anything, any organism that is too small to be seen with the naked eye. So you have to use a microscope in order to view this organism. So this is largely bacteria, but also in this category, we'll be talking about the viruses, we'll be talking about the fungi, the protozoans, which are some of the parasites. We'll talk about prions, and we'll be talking about a wide variety of different types of microbes. So these are all microbes that we'll be talking about. So going back to decomposing organic waste, there's something else that we need to talk about is the term organic. Most people, when they hear the term organic, they think of the organic section at the grocery store. Okay, that is not what organic means here. Organic means, in a scientific context, that it contains carbon. So again, anything organic means that it contains carbon at the molecular level. Okay, so decomposing organic waste. So all these microbes, particularly the bacteria and the fungi, they work by decomposing organic waste. So let me give you a definition or an example of this organic waste. Organic waste includes basically every living organism. Every living organism contains carbon. And so when we have something that dies, such as tree limbs or leaves or grass or even an animal, when it dies, we have bacteria and we have fungi that kind of take over. They consume that organism, they break the elements down, the nutrients down to the elemental level. The elements are then recycled in the earth and used to create new organisms. So these microbes, particularly the bacteria and the fungi, are very, very effective at decomposing organic waste. Next, performing photosynthesis. This is a big one. Normally, when you think of photosynthesis, you think of plants and trees and how they use the sun to make their own energy. And that is all correct and true. However, what you may not know is there are large categories of bacteria that are able to perform photosynthesis. So they are able to use sunlight to make their own energy. And they actually give off a combination of oxygen as well as carbon dioxide. There was one study that suggested that the majority of the oxygen that humans consume is actually a waste product of the bacterial photosynthetic process. So they're saying that bacteria produce more oxygen than trees do. Okay. All right, the next one, the third bullet, producing ethanol, acetone, vinegar, cheese, bread, other things of that nature. So ethanol, this is drinking alcohol. Okay. So anytime you have drinking alcohol, fermentation has been used. Usually bacteria, sometimes yeast, is used to allow sugars to undergo fermentation to wind up with ethanol as an end product. Acetone, this is stuff like paint thinner and things like that. These are also byproducts of bacterial waste. Vinegar. So vinegar is acetic acid. Again, these microbes, a lot of these microbes, they produce waste products just like humans do. They just produce a different variety of waste products. So some of these bacteria literally produce ethanol or acetone or even vinegar as a waste product. We as humans have figured out how to collect the acetone, the vinegar, the ethanol, the waste products of these bacteria and repurpose it and be able to resell it. Cheese, okay? So a good example of where bacteria and yeast are used in cheese. If you're familiar with blue cheese, you either love it or you hate it. Blue cheese has a very strong smell and very strong taste, and if you look at it, it has a blue line running through it. Well, what is the blue line from? Literally, again, it's a combination of bacteria and mold that's been added to the cheese. The bacteria and the mold are literally eating the cheese and producing waste products. The waste products is what gives that cheese its tart tanginess. Okay. So I was watching a show the other day and it talked about the most expensive cheese in the world. It had a very thick covering of mold growing over the cheese. So you had to take this really big knife and cut through like an inch layer of mold to get to the cheese. Okay. Generally, cheeses, the stinkier the cheese, the reason it stinks is because certain strains of bacteria, yeast, fungi have been added to that cheese to give it its distinct tart, tangy taste and its unique, usually pungent or really strong odor. Bread. This is another very good example of where microorganisms play a role. There is a yeast. So yes, yeast are part of the fungi category. The scientific name for this yeast is called Saccharomyces cerevisiae. Okay, Saccharomyces cerevisiae. You will see it spelled in a later chapter. You don't have to worry too much about the spelling of it for right now. But this is also known as baker's yeast. So if you've ever baked bread, or if you're familiar with the process, you have your bread ingredients and then you have to add baker's yeast to it. And typically you have to activate the yeast with warm water. The yeast is in a dehydrated state. It's not doing anything, but once you've activated it with warm water, it rehydrates the yeast. The yeast start producing gas as a waste product. The gas is what causes your bread to rise. So if it wasn't for this yeast, Saccharomyces, all of our bread would be pancake flat. Okay. It is again due to this yeast which allows all of our bread products to rise. One not even mentioned on here is yogurt. Okay. I eat yogurt almost every day. If you look at the container of pretty much any yogurt on the market, it will tell you that this yogurt contains active live cultures. Well, active live cultures of what? If you go on and read on the yogurt container, it will tell you the particular species of bacteria that have been added to that yogurt. One of the main benefits of this is that bacteria often produce something called lactic acid, which is what gives yogurt its tart, tangy taste. Okay. Then there are other yogurts on the market, such as Activia yogurt, that have additional strains of bacteria added because it's supposed to promote good gut health. Okay. The topic of probiotics, which we'll talk more about in a later chapter, has really taken off over the past few years. It's basically living strains of bacteria that you consume on purpose because there is research that suggests probiotics help the gastrointestinal tract. It helps relieve things like constipation. It helps relieve diarrhea. It helps relieve gas and bloating, and overall is supposed to really promote again, like I said, good gut health. Okay, another topic in which microbes help us is by producing insulin and many other drugs. The story behind insulin is actually really, really interesting, because this is where science meets technology. We have bioengineering, we have these new products that are being bioengineered through genetic engineering or genetic modification. And basically what's going on here is at one point in time, when a patient needed insulin, the insulin was derived from pigs. We used pig insulin to treat people. Okay. The problem with that was an unstable insulin supply, and what would happen if the if pigs were infected by some type of disease or something of that nature, what would happen? And so scientists were actually able to take E. coli cells, insert the gene that produces insulin into these E. coli cells, and today 99.9% of all insulin is made in these genetically modified E. coli cells. Okay. Other drugs. The first drug ever discovered on accident was penicillin, or the first antibiotic, I should say. Penicillin has saved many, many lives. It was first discovered in 1928. It turns out, though, that it's produced by a fungus. And this fungus, Penicillium chrysogenum, produces what we call penicillin today. And as the semester progresses, again, we'll talk about other drugs that have been discovered, sometimes by accident, by some of these microorganisms, these fungi and these bacteria. Lastly is the topic of bioremediation. Bioremediation is the idea of cleaning up the environment without introducing harmful chemicals. Now, if you were to go on to the next slide, there's a picture of bioremediation. I cannot switch between slides because it messes up my recordings. However, on the next slide, there is a picture of an oil spill that is being cleaned up by adding certain strains of bacteria to the water. This is new. Oil spills in the past have always been very costly and very expensive to clean up. Very time consuming to clean up, okay? And usually chemicals were added to the oil spill. Well, back in around 2010, there were some species of bacteria that were discovered that if you added the bacteria to the oil, the bacteria actually ate the oil. They were able to use the oil as a food source. And they would eat the oil, they would break down the oil, and then they would convert that oil to harmless byproducts such as water and gas, and so harmless water and gas was released into the environment. So that again is an example of bioremediation. You're using some type of microorganism that's already present to clean up the environment so you don't have to introduce potentially harmful chemicals. Okay. All right, moving on to how microbes harm us. This is usually what we are hearing about in the news. This is usually what we're familiar with. How do microbes harm us? Well, yes, we know that microorganisms cause a wide variety of disease. They cause plague, they cause tuberculosis, pneumonia, swine flu, hepatitis, rabies. They cause regular flu. They cause chickenpox that we just came through a COVID outbreak. So there are so many different diseases that are caused by a variety of microbes, whether it's a virus or a bacteria or a fungi or even some of the parasites. Also, these microorganisms harm us by causing food spoilage. So say you cook some spaghetti and you forget that you cook it, you put it in the refrigerator, you forget that it's there, and then 2 weeks later you go back and you find this leftover spaghetti. If you look at the spaghetti, it will oftentimes have a layer over the top of the spaghetti. That is known as a biofilm. Okay. That is a combination of bacteria from the environment that has landed on the spaghetti, has been able to use the spaghetti as a food source. It replicates, the bacteria are able to replicate because they have an unlimited food source. And they produce waste products that can be toxic if consumed. So that is why you never eat food that has spoiled, whether it's out on the counter or whether it's been stored in the refrigerator. If it's spoiled, if it's been there for a while, you never eat it because bacteria reproduce so quickly that it only takes a few hours for millions of bacteria to potentially be there. And we will look at the math associated with bacterial division in another chapter later on. Okay. All right. So that's everything about that slide. If we move on to, okay, so here's the slide that I was talking about with bioremediation, where the oil spill was cleaned up by a couple of particular species of bacteria. Okay. All right. Let's go back to the 1700s and let's talk about this famous guy. He went by the name of Carolus Linnaeus. We give him credit for establishing the scientific nomenclature system in the year 1739. Now, I am not big on dates, okay? Yes, you need to know who the person was and what he did, but you don't really have to know exact dates because in chapter 1, there are a lot of dates, okay? So basically back in this time period, though, there was no electronics, there was no electricity, there was no communication, there were no phones. If you communicated, it was by horseback or by boat. A person had to carry a message. Well, if you went to another country by boat or by horse, there was a very good chance that the people there spoke a completely different language. And so Carolus Linnaeus said that all living organisms needed a scientific name, so that no matter where in the world you went, if you said the scientific name, everyone would understand what you were talking about. So this is where each organism has two names. The prefix bi means two, so binomial nomenclature, two name system. The genus is the first name, the specific epithet is the second name. Now the specific epithet is sometimes known as the species. If you notice, the genus is always capitalized, the specific epithet is never capitalized. If you are handwriting this, then it needs to be underlined. If you are typing this, then it needs to be in italics. Okay. Some examples I have on the next slide. We'll go over this in a minute again on the next slide. A lot of these names are used when they were naming these organisms. They were named to describe either what the organism looked like or maybe the scientists that discovered them. And there are still new organisms being discovered on a weekly basis. There's one estimate that said we've only actually discovered about 10% of the species that actually live on planet Earth. Okay. So let's look at some examples of the scientific nomenclature system. Here in the upper left-hand corner, we have a picture of Staphylococcus aureus. Very, very common bacteria. You will a lot of times hear me simply call it staph. Okay. The prefix staph means cluster of grapes. Make sure you know that. Staph means cluster of grapes. If you look at it, the picture, those bacteria, they're clumped together. They do kind of look like clusters of grapes. Well, if you'll notice that Staphylococcus is capitalized, aureus is not capitalized. However, the entire thing is in italics. Okay. The next term, Escherichia coli. Now, in the news, you typically hear it called E. coli. This is one of those bacteria that gets a bad rap. We'll talk a lot about E. coli this semester. Every person in this world just about has E. coli in their intestines. Okay. There's good E. coli and then there's bad E. coli. So as far as E. coli is concerned, you see a picture of E. coli in the upper right-hand corner. Now, I will say that E. coli is not normally green. This E. coli has been tagged with a fluorescent tag, which we'll talk about in the next chapter when we talk about microscopes. Okay, before I move ahead, you see that the E in Escherichia is capitalized. The C in coli is not capitalized. The entire phrase Escherichia coli is in italics. The last one is Streptococcus pneumoniae. Okay. First of all, you notice it meets the rules. The S is capitalized, the P is not capitalized, the entire thing is in italics. Now, here's what you do need to know. The prefix strep means chain of cells, because if you look at the bottom left corner, that is a picture of Streptococcus pneumoniae, and you see that it looks like a chain of cells, or a chain, okay? So make sure you know those two prefixes. You will see them again. And then in the bottom right-hand corner, this is the guy that discovered E. coli. You do not need to know anything about him. I just thought it would be nice to put his picture there. Okay, here's a list of things that we will be studying this semester. All right, so when you think of microbiology, you probably think of bacteria, and that is correct. About 80% of the material that we talk about will be bacteria. But we'll also talk about the archaea. Now, what's interesting about the archaea is this group didn't exist until 1978. Up until 1978, it was thought that archaea were bacteria. We'll talk more about that in just a few minutes. So yeah, we'll be talking about the archaea. We'll be talking about fungi. Okay, so we'll be talking about the yeast as well as the molds. We'll be talking about protozoa. Okay, here's another term. The prefix proto means one. Zoa means animal. Protozoa. These are one-celled animals. Okay. An example includes something like the amoeba or the paramecium. Okay. We will talk about several others. These are actually very fascinating. We'll talk about several others later on in this semester. We will very briefly talk about algae. There's just a couple of algae that are considered to be microorganisms that can potentially cause disease in humans. Viruses are another one. It's a big one. We have a whole chapter devoted to viruses. So yes, we'll be studying viruses in this chapter. Multicellular animal parasites, also very fascinating. These can get very, very large. I'm talking 30 feet in length. Examples include some of the tapeworms, like the beef tapeworm, the pork tapeworm, the fish tapeworm. The reason that we study them in microbiology, okay, because they do get large enough that you can see them without a microscope. But the reasons that we study them in micro is because a lot of times they have very complex life cycles. They'll have 13 to 15 stages of their life cycle. So they're typically studied here because they do cause human disease and disease of other animals. Also because they have such complex life cycles, the majority of their life cycle stages, they're microscopic. And so that's why we study them in micro. And then lastly, also very interesting, I think, are the prions. This is one of the reasons that I went into microbiology. I really do find these topics to be fascinating. Prions were not discovered until 1982. What are they? Well, they are nothing more than a protein, a brain protein, okay? A brain protein that randomly mutates. There's still a lot that we don't know about prions. But probably the most interesting thing about prions, they have a 100% death rate. Nobody has ever survived a prion disease. And yes, we will talk about those as well. An example of a prion disease is mad cow disease, in case you were wondering. There's only a handful of diseases that are said to be caused by prions, and we'll talk more about those. They're actually lumped in with the viruses. So when we get to the virus chapter, at the end of that chapter, we'll talk about the prions. All right, let's talk a little bit more about some of these in depth. So let's talk about bacteria first, because again, very large category. All right, so before we even start talking about the bacteria, the first word, prokaryotic. Now you should know this word. You should have learned it at some point, hopefully in A&P. So there's two cell types. You have prokaryotes and you have eukaryotes. Okay. In anatomy and physiology, you learned about eukaryotes. You learned about cells with a nucleus. You learned about cells with all the typical organelles that you typically learned about, like a mitochondria and rough and smooth endoplasmic reticulum and a Golgi. Okay, well, the thing about prokaryotes are very, very simple. Prokaryotes include the bacteria and the archaea. So this is one area that some students find challenging, is because we are talking about a completely different cell type than what you are probably used to studying. Prokaryotes are extremely small, okay? At low magnification under the microscope, they look like specks of pepper. That's about it. Prokaryotes don't have a nucleus. Prokaryotes don't have most organelles. Prokaryotes do have a cell wall, okay, made of something called peptidoglycan. Long word, it sounds weird. Become familiar with it though, because you will hear it numerous times. It is called peptidoglycan. Okay. So bacteria have peptidoglycan. You will not find peptidoglycan in any other organism on the face of this planet. It is found solely in bacteria. You might want to know that. That is a key important detail of the bacteria. Okay. Bacteria reproduce by binary fission. There is no mitosis. There is no meiosis. Okay. Binary fission is a very simple process. It can take as few as 20 minutes. Literally, one cell becomes two cells. One cell will elongate, the chromosome is copied, and then the one cell gets cut in half. Each new cell has a complete copy of the chromosome. Binary fission is considered to be asexual reproduction because all of the offspring are genetically identical to the parent. There was never any exchange of genetic material. Okay. So again, binary fission is an example of asexual reproduction. They do have, again, the bacteria do have a cell wall. If you look at the picture in the upper right-hand corner, you see that the mitochondria of a cell, of a eukaryotic cell, is about the same size as a bacteria. So it makes sense that bacteria can't have mitochondria because they're the same size. So again, these bacteria that we're studying, very, very different. If you look at the picture at the bottom left corner, this is a typical bacteria cell. Nothing fancy, no nucleus. Okay. Again, most of the organelles are missing. The only organelles that you'll find are ribosomes. All right, let's talk about how they get their energy. So how do they get their energy? Well, there's a few different ways in which bacteria get their energy. And when I'm talking about getting energy, I'm talking about making ATP here, making adenosine triphosphate, which you learned about in A&P. Okay, some bacteria, actually a lot of bacteria, are very similar to us. They eat organic chemicals, okay? Because if you look at all the foods that we eat are organic. If you look at the chemistry definition of organic, remember the chemistry definition of organic means it contains carbon. Okay, if you were to look at fats, all fats have carbon in them. If you were to look at protein that you eat, well, what is a protein? Protein is nothing more than amino acids. If you look at all amino acids, there's carbon there. If you look at carbohydrates Carbohydrates are complex sugars. All sugar has carbon. So again, all the foods that we eat, according to the scientific definition of organic, all the foods that we eat as humans are organic. Okay. Bacteria are able to get their same energy. Now, they don't have a mouth per se. They get a lot of their nutrients through absorption. But they're able to absorb a lot of these same chemicals or these same elements that we are. So they're able to get energy from fats and carbohydrates, lipids, sugars, amino acids. So that is what is meant by organic chemicals. Now, there are a few bacteria that are very weird, very unusual. They are able to get their nutrients from inorganic chemicals. This is far more rare. Inorganic chemicals are things that are chemical components that do not have carbon, such as calcium, magnesium. Some bacteria are able to use heavy metals to get their energy. They're able to eat metal. There is a very unique bacteria that is able to eat rock and get their energy from eating rock. Okay, so that is inorganic. It lacks carbon. And then we talked about how some bacteria are able to undergo photosynthesis, so they don't have to eat anything. They're actually able to use sunlight to make their own ATP. Okay, so the next group of microorganisms that we'll be studying this semester are the archaea. Now, this is the interesting group that didn't exist until 1978. The reason this group didn't exist till then is we thought this group belonged to the bacteria. So the archaea didn't exist because they were grouped in or they were lumped in with the bacteria. Because they're the same size as bacteria, they're the same shape as bacteria, they behave very much like bacteria, so it was thought that they were bacteria. However, in 1978, what happened was we were able to do genetic sequencing for the first time. Based on their genetic sequence, it was found that this unusual group were not bacteria at all. They were actually more closely related based on genetics to eukaryotic cells, such as ourselves. So they were kicked out of the bacteria group and they were given a new heading. They are now known as the archaea. They are prokaryotes like the bacteria are. However, they do not have peptidoglycan. And they tend to live in unusual environments or extreme environments. For example, some include the methanogens. These are archaea that produce methane gas. Some are extreme halophiles. Halo means salt. Phile means lover. These are salt lovers. Okay? And then some are extreme thermophiles. Therm meaning heat. Again, phile means lover. These are heat lovers. They are found in really hot environments. And then lastly are the extreme cryophiles. The prefix cryo means cold. Phile means lover. So cryophiles are your cold lovers. These are ones that can be found in really cold environments such as the North Pole or down south in Antarctica. Okay. The next group of microorganisms that we'll be talking about are the fungus, or fungi is plural. We have now officially switched cell types. We are now talking about eukaryotes. So this is what you studied when you took anatomy and physiology. Eukaryotes do have a nucleus. They do have traditional organelles. Okay. With the fungi, they do have cell walls made of chitin. Chitin is somewhat unusual. The only other place that it's been found so far is in the exoskeleton of certain types of shellfish. They use organic chemicals for energy. So again, the term organic chemical basically refers to the same things that we eat. That's why if you buy a loaf of bread and you let it sit on the counter for too long, mold will grow. That's because the mold has been eating the bread. It's been using the nutrients from the bread to replicate and to grow. And that's why the mold eventually grows large enough that you can see it without using a microscope. Now, in microbiology, we'll basically be talking about the molds and the yeast. We are not going to be talking so much about mushrooms because mushrooms tend to be macroscopic, meaning you do not need a microscope in order to see the mushrooms. So again, as far as molds are concerned, you can see a picture of a mold in the bottom right-hand corner. Okay. Molds usually consist of two parts, the mycelia and the hyphae. So mycelia, if you look at the picture in the bottom right, the mycelia is that dense mass in the upper right-hand corner. And the hyphae is that white part. It looks like it's actually these really fine hair-like projections. I remember it because hyphae, hairs, they both start with the letter H. Okay. And then alongside the molds, we'll be talking about the yeast. So again, yeast, this is responsible for causing bread to rise. We talked about baker's yeast. We talked about Saccharomyces cerevisiae, the scientific name for baker's yeast. We'll be talking about other yeast infections such as thrush later on when we get to the fungus chapter. But yeast are typically unicellular. You do have to have a microscope in order to see them because they are only composed of one cell, and they are very important. So that is basically everything that you need to know from this particular slide on the fungus and the yeast. Okay, so this next slide talks about the protozoans. You can take that word and break it down. Proto means first, zoa means animal, first animal. It was thought that these were the first animals that were in existence. They are single-celled, so that means the entire animal only consists of one cell. They are microscopic, but examples include things like the amoeba, which you can see here in this picture, or the paramecium. So these are eukaryotes. They're a very wide branching, there's many different types of protozoans that fall within this category. As you see in the picture here of the amoeba, it does not have a mouth. So it would get its nutrition through absorption. However, some of these protozoans are more advanced. They do have more of a mouth-like structure. And in that case, then they would actually be able to ingest the organic chemicals. But once again, organic chemicals, these are the same type of foods that we eat. So they basically need the same type of nutrition that we do. I would say 90 to 95% of the protozoans are found in water. Therefore, they are swimmers. Therefore, they need appendages to help them swim or to help them move through water. Now, I'm sure you talked about cilia and flagella in anatomy and physiology. So flagella are longer and a little thicker. Cilia are really fine hair-like projections. So those are probably review for you. What may be new for you, though, is the term pseudopod. The prefix pseudo means false. Pod, which is short for podia, means foot. So pseudopod literally translates into false foot. And you can see the pseudopods are labeled on this picture here. That's because the amoeba has control over these pseudopods to allow them to use those to help propel them through that watery environment. Okay. Moving on to viruses. Viruses are very unique for several reasons. One of the main reasons in which they're unique is that they are acellular. They are not composed of cells. They are not prokaryotes. They are not eukaryotes. They are acellular. So that prefix A means without. So acellular literally translates into without cells. So these viruses are not composed of any type of cell. Another very unique characteristic of the viruses is that they have DNA or they can have RNA. So what you've learned all your life is that all living organisms contain double-stranded DNA and single-stranded RNA. For living organisms, that is correct. viruses are a little bit different. We'll talk about that in a minute as to whether viruses are considered to be alive or not. But there are some viruses that have double-stranded DNA, like we do, but there are also some viruses that have single-stranded DNA. There are some viruses that have double-stranded RNA, which is, again, very unusual. But then there are some viruses that have the single-stranded RNA, like we do. Okay, we'll talk more about genetic components of viruses when we get to the virus chapter. Structurally speaking, viruses are very, very simple. A lot of viruses are composed of two parts. The first part is some type of genetic material, whether it be DNA or RNA. The second part is a protein coat. The purpose of the protein coat is to protect the genetic material, to protect it from the environment, protect the genetic material from being broken down. Okay. Now, some viruses are a little bit fancier. They have an extra layer around them known as the lipid envelope. Okay. So in one of those viruses, there would be three components. You would have your genetic material, and the genetic material would be surrounded by a protein coat, and then the protein coat would be surrounded by a lipid envelope. Okay. This becomes important when we talk about destroying viruses, particularly destroying viruses with hand sanitizer. Hand sanitizer is only effective on viruses that have a lipid envelope around them. If the virus does not have an envelope, hand sanitizer does nothing towards the virus. Now, luckily, COVID did have an envelope around it. The human flu virus has an envelope around it, so they are inactivated by hand sanitizer. However, if we were ever faced with an outbreak of a virus that did not have an envelope around it, your hand sanitizer would not work. It would not be effective towards the virus. Now, like I mentioned, there's long been this great debate over asking the question if viruses are alive or not. Okay. Viruses were first discovered in 1890, and this question has been asked numerous times. It is the general consensus of the scientific community, though, that viruses are not said to be living. And there's two main reasons for this. One, viruses are acellular. Well, in a minute, we'll talk about the history of micro, and I'll talk about Robert Hooke and the cell theory, which simply states all living organisms are composed of cells. Well, viruses are acellular, so that goes against the basics of cell theory. But the last bullet is also very important in proving that viruses are not considered to be living, and that is a virus is only active if it is attached to an appropriate host cell. If you were to look at a single virus particle and you were to watch it under the microscope, it would appear to be dead. It's not doing anything. It's not growing. It's not replicating. It's not reproducing. It's not performing respiration. It's not producing waste products. It's literally doing nothing. It has to be attached to an appropriate host cell before the virus is activated. For that reason, it is said that viruses are not considered to be living. Okay. Now, if you look at this picture, if you look at the slide, there is a picture of a T-even bacteriophage on this slide. We will talk more about bacteriophages later on, but just to give you an introduction to a bacteriophage, these are viruses that infect bacteria. So bacteria are not immune to viral infection. Something else that's kind of interesting, there are said to be viruses that can infect other viruses. Just keep in mind the general consensus is that they're not living. And when we're watching the news or something like that, we may hear the phrase, oh, we got to kill the virus. Well, you can't really kill a virus because you can't really kill something that isn't living in the first place. The best we can do is inactivate the virus. So that's actually the correct terminology that needs to be used. We're not killing the virus. We need to inactivate the virus. Okay. Moving on to this group, these are the multicellular animal parasites. Okay. They are eukaryotes. Therefore, they do have a nucleus as well as traditional organelles. They are multicellular, so they can get very, very large. I'm talking some of these can easily be 30 feet in length. Okay. So within this category are the helminths, the term in green. Helminth means parasitic worm. Not all worms are parasites, though. Therefore, not all worms are said to be helminths. For example, an earthworm. They're not human parasites. Therefore, an earthworm is not considered to be a helminth. However, when we talk about things like the tapeworm, or we talk about the pinworm, or we talk about ascaris, or we talk about heartworms, those are examples of helminths because those are considered to be parasitic worms. The main reason that we study them in microbiology, because they can get very large. You do not need a microscope in order to see all of them. But the reason that we tend to study them in microbiology is because they have very complex life cycles with 13 to 15 stages in their life cycle, and the majority of those stages, they're microscopic. So for that reason, along with the fact that the helminths are human parasites, they're classically studied in an entry-level microbiology course. So here you can see the pictures. At the top, we have a tardigrade, also known as a sea bear. Okay, you can see the little teeth on the tardigrade. And then below that, you see a heart, and you can see heartworms. Okay. Now this looks to be a fairly severe case of heartworms. You can see there's clusters of worms coming out on the right side. You can see they almost look like strands of spaghetti. But you can see these worms, and typically when you think of heartworms, because they're carried by mosquitoes, heartworms are traditionally found in animals like cats and dogs. However, there have been human cases of heartworms. All right, let's talk a little bit about the classification and the organization of organisms. All right. So it all started with Charles Darwin, who was very famous for his theories of evolution. But he basically said that all living organisms could be divided into two kingdoms. Those two kingdoms were plants or animals. Okay. Well, as our knowledge of science grew over the years, we quickly discovered that there were organisms that existed that clearly were not plants, and they clearly were not animals. So that's why other groupings to the kingdoms became in existence. So we added the fungi. So that is now a third kingdom. We added the protistans. These are those protozoans, those single-celled animals. And then we added the Monera. Okay? And so that is why today we have 5 kingdoms. The 5-kingdom system was great. It's still taught today. It's still used today. But the problem with the 5-kingdom system goes back to 1978 once again. Once we discovered the archaea, there is no place in the 5 kingdoms for the archaea to exist. So if we move forward, scientists went back to their drawing board to try to come up with another organizational scheme to include the archaea. Because the 5-kingdom system was great, but again, it excluded the archaea. So that's when scientists discovered, or they made, the 3-domain system, which is also still used today. So the domain on the far left, those are your bacteria. The domain in the middle, now we have the archaea that are included. And then if you look at the domain over on the far right, you have the eukarya, which is everything else, your plants, your animals, your fungi, so on and so forth. Okay, so 5 kingdoms, 3 domains. And if you look at the archaea, remember I said earlier that The archaea look like bacteria. However, based on genetic sequence, they're actually more closely related to the archaea. To the eukarya, and you can see that in this slide. The archaea branch off of the eukarya, not the bacteria. So again, this is proof that the archaea are more genetically related to us than to the bacteria. All right, so now let's switch gears a little bit and let's talk a little bit about the history of microbiology. So as far as the beginnings are concerned, it is thought that bacteria were among the very first life forms. Although there is now some evidence that suggests that viruses were actually here before bacteria were. However, let's go ahead and jump ahead to the year 1665 and Robert Hooke. Microbiology did not exist as a science yet. It was not a discipline of science. In fact, science was still in its fairly early stages. So the people that we're about to talk about were not scientists. For example, Robert Hooke, he was not a scientist. He was a monk. He lived in a monastery. And he had a very simple magnifying glass that he would take to outside with him, and he would look at different things like leaves and grass and various things under his little magnifying glass. And he drew recordings of everything that he saw. And he looked at cork slices. That's one of the things he's really famous for. But he also looked at leaves. And what he noticed was that there were these organizational patterns. We now call them cells. They were not called cells at that point because nobody had seen them before. So they didn't have a name. But Robert Hooke lived in a monastery. And in a monastery, each bedroom was labeled a cell. And the bedrooms were lined up in an organizational pattern that reminded him of the leaf pattern. And so he termed each square or each box of the leaf a cell. So he is the first person to ever use the term cell in a scientific context. He went on and he created the cell theory, which simply states all living organisms are composed of cells, which today we already know this. We've known this for hundreds of years. But for this particular point in time, this was considered groundbreaking. Okay. Now, moving ahead a few years to 1673, we have another person by the name of Antony van Leeuwenhoek. Again, he was not a scientist, he was a draper, he worked with fabric. Well, he wanted to create a magnifying lens so that he could look at his fabric, because the better his fabric was made, the more he could charge for the fabric. Well, it turns out that he had this really uncanny ability for creating magnifying lenses. He was really good at it. And he created a magnifying lens that was able to magnify 40 times. Which was considered great for this particular time period. So he also went around collecting various things to look at under his primitive magnifying glass. He looked at plaque from his teeth, he looked at fecal matter, he looked at rainwater, he looked at pond water. Well, one of the things was, when he was looking at pond water, he noticed that there were these little critters in the pond water moving around. And he didn't know what they were. They had, again, never been seen before. So he gave them the name animalcules, which means little animal. Because, again, he didn't know what they were. Now, today, we know what he saw were the protozoans. He was looking at those single-celled animals, such as the amoeba and the paramecium. Okay. We also give him credit for developing the first primitive microscope. As you can see here in this picture, it's that gold structure on the right. You use it by, you place your specimen on top of the screw on the tip, and then you hold that gold flat apparatus up to your eye. There's a lens in there. The lens is ground to magnify whatever's on the tip of that screw 40 times. And this was his primitive microscope, so we give him credit for developing one of the first microscopes. He also recorded all of his drawings. He published a book. Some of the books are still in existence today that showed all of his drawings and his recorded notes. The thing about Van Leeuwenhoek, though, is that when he died, he kind of took his secrets to the grave with him. He kept no written record book, and he didn't tell anybody his secrets for grinding lenses. And so when he died, he took his secrets to the grave with him. Now, science still continued to develop without him. We still made advances in microscopes. However, we weren't able to replicate his exact experiments until the 1950s. Okay, so the transition period between spontaneous generation and biogenesis. Aristotle was a philosopher around 400 BC. He's very, very famous. People today have heard of Aristotle. He had a lot of great ideas. However, this was not one of them. He's the one that originally proposed the idea of spontaneous generation. So the idea of spontaneous generation is that something non-living can somehow create something that is living. Okay, so for example, if the question was asked, where do alligators come from? Well, according to spontaneous generation, the alligator is typically found in water. Well, there's usually trees found near water. So when the tree died and it fell over into the water, somehow the tree itself turned into an alligator. Okay. Or another example is, how did frogs come to be? Well, frogs are typically found near mud. So it was thought during those days that the mud itself somehow turned into a frog. Another common thought or belief, which we'll talk more about, it'll make more sense on the next slide. So if the question, okay, where do maggots come from? Well, remember, we're in a time when there's no refrigeration, so food would often spoil, food would go bad, and usually maggots would appear. It was thought that somehow the decaying meat or the spoiled food turned into the maggot. So something non-living could somehow generate and create something that was living. There were actually recipe books for that time period on the idea of spontaneous generation, on creating something for nothing. If you wanted bees to appear, you could use dirty clothes to summon bees, and somehow the dirty clothes would turn into bees. Shakespeare actually wrote a couple of these recipe books on how to create something from nothing. This was everybody's beliefs. Nobody questioned it. It was a topic that had been passed down from generation to generation. It was not questioned. So it wasn't until much later, the 1800s, that we were finally able to disprove that spontaneous generation occurred. And the opposite of spontaneous generation, which is biogenesis, so to get the belief that biogenesis is actually what occurs today. So biogenesis is basically stating that you have something living that creates something else that's living. So living can only come from other living organisms. You cannot take something that's dead or inanimate and somehow create a living, breathing creature. However, it took a few hundred years and numerous experiments between Aristotle's spontaneous generation idea and the whole idea of finally disproving spontaneous generation and believing in biogenesis. So let's take a look at some of those historical experiments. So in 1668, we have a guy by the name of Francesco Redi. We give him credit for being one of the very first to record accurate record. We give him credit of being the first to display experimental science, because of whenever he did any type of experiment, he recorded his notes in great detail so that his experiments could be replicated, and his account or his historical account, is some of the first records that we have of where an experiment actually took place. So he took 6 jars, filled all 6 jars with decaying meat, and according to him, and according to the theory of spontaneous generation, well, the decaying meat should have somehow turned into maggots. But the jars, he covered 3 of those jars with a fine net, and the ones that were covered with a fine net, there were no maggots that appeared. However, the other 3 jars that were open to the environment, well, maggots appeared. According to the theory of spontaneous generation, decaying meat would result in maggots. So why were there 3 jars that were covered with fine net? Why didn't they have maggots? So we are starting to, very early experiments, trying to get away from the idea of spontaneous generation. So if we move ahead to the next guy, John Needham, if you look at the year, we're now in the middle of the 1700s. Unfortunately, this was kind of a setback. He also didn't believe in the idea of spontaneous generation. He was like, wait, it just doesn't sound right. He tried to create an experiment also, but he got some of the conditions wrong. Okay. He took nutrient broth, which would be something like chicken stock or beef stock. He heated it and then placed it in a sealed flask, but there was microbial growth. He didn't think that there would be microbial growth, but the characteristics of the experiment that he got wrong was, we know that there's bacteria everywhere, and bacteria landed on the flask. Bacteria landed inside the flask. The flask itself was not sterile, and that's why there was microbial growth. Okay. But if we move ahead to 1765, we have an Italian by the name of Lazzaro Spallanzani who finally got the conditions correct. So he took nutrient broth, placed it in a flask, heated the entire thing, sealed it. There was no microbial growth. So we are once again on our way of moving forward to disproving the idea of spontaneous generation. But now if you look at the year, we're all the way up to 1861. We have a very famous guy by the name of Louis Pasteur, and I'm sure everyone's heard of Pasteur. He believed that there was something in the air. We know now he was correct. He was thinking bacteria, but he was faced with a lot of critics. There were a lot of people that did not think bacteria existed. He also did not believe in the idea of spontaneous generation. He was trying to prove that spontaneous generation did not exist. And he was also trying to prove that there was something in the air that was causing microbial growth to occur. So he set up 2 separate experiments. You can see the setup here on the slide. He had nutrient broth placed in flask, heated, not sealed, versus nutrient broth placed in flask, heated, then sealed. The ones that was not sealed, well, it was open to the environment, so of course there was going to be microbial growth. But he said, you see here, these flasks that I have sealed, there was no microbial growth. So to him, this was proving that there was something in the air, that there was some type of something that he couldn't see that was present in the air. Okay. But he faced critics, and his critics stated that the reason there was no microbial growth was not because it was sealed, but because in order for microbial growth to occur, the experiment itself needed to be open to fresh air. Fresh oxygen needed a way to enter into the experiment. And so this is where he set up an experiment, and he created his very famous swan-necked flasks. Here you can see the S-shaped flask, or the swan-necked flask. He created these himself. So basically he took a flask with a very long, thin glass neck. He poured the liquid into the opening. And if glass is really thin, you can heat it up and you can bend it. being careful, of course, not to break it. And that's what he did. He heated up the neck and he bent it to create the swan neck, or this S shape. And he heated the liquid in the flask over an open flame. It killed all the bacteria. Well, he kept it open for a week, maybe 2 weeks, and the liquid contents of the flask stayed sterile. To him, once again, this was proving that there was something in the air and that you could create an experiment, but you had to do so carefully in order to keep microbes out. So he created an experiment that was open to fresh air, but that did not allow microorganisms into his S-shaped flask. Because what would happen if microorganisms entered the opening of the flask, they would get caught in the crook of the neck and they wouldn't be able to go back up, to go back down, to fall into that sterile liquid. So to him, he was proving that yes, there was something in the air that existed, and also disproving the idea of spontaneous generation. Because if we move ahead to the next slide, you will see that the golden age of microbiology became or ran through about 1857 all the way up through 1914. There was a lot that happened during this time period, which is why it's called the golden age of microbiology. We discovered bacteria. We discovered that bacteria cause disease, because even after bacteria were discovered, it still took quite a while for them to realize that it was bacteria that actually caused disease. We also discovered viruses in this time period. We discovered our first drug during this time period. We made advances in microscopes and advances in technology during this time period. And so a lot was accomplished during this time period. And this is when microbiology officially became established as a science or as a branch of biology. Now, Louis Pasteur, we officially give him credit for disproving spontaneous generation, but he also worked a lot with wine fermentation. He wanted to know how yeast and bacteria worked to create wine. He also wanted to know why wine went bad if left uncorked. Because if left uncorked at room temperature, wine turns into vinegar, which is acetic acid. He wanted to know why. Wine and other types of alcohol were extremely common in Europe. It was oftentimes safer to drink alcohol than it was to drink water. If you drank water, you had a really high chance of getting sick and even dying from it. However, if you drank alcohol, the fermentation process of alcohol kills bacteria. If you drank alcohol, even weak alcohol, you had a much higher chance of not getting sick. So that's why Europe, even today, has a very high alcohol consumption, because it's all of historical basis. Even little kids way back in the day would drink very dilute alcohol because it was safer to drink alcohol than it was to drink plain water. Louis Pasteur also created the idea of pasteurization. So dairy farms have long been a home to a variety of different pathogens that are associated with dairy products. And we'll talk more about this later on in another chapter. But a lot of people would drink dairy products and then they would get sick and they would die from it. Because we're still in an era where there's no antibiotics. So what he started doing was a simple process of pasteurization. This was a process where you would heat dairy products to kill the majority of the bacteria. Now, pasteurization is not sterilization. So if you buy milk and you never open the milk will still go bad because there's living bacteria in the milk. However, pasteurization is designed to kill off about 90 to 95% of the bacteria that's present. So most of the bacteria that's present. And not only milk, but most of our dairy products, the milks used to make cheeses, there are some fresh fruit juices that have all underwent pasteurization, making it shelf stable or making it stable in refrigerated conditions for a longer period of time. Louis Pasteur was also the one that first correlated the bacteria to causing human disease and infection. So again, Louis Pasteur did quite a bit in the area of microbiology. Okay, so now let's take a look at medical microbiology. Let's talk about the first guy, Ignaz Semmelweis. The year was around 1840. So imagine you're female, it's time for you to give birth, you're going to the hospital to give birth, the doctor comes in to deliver the baby, he has not washed his hands, he's not wearing gloves, there's no type of sanitation. Well, that's exactly what it was like to give birth in a hospital prior to around 1850. Ignaz Semmelweis, he was a physician and he delivered babies for a living. And basically, you had a very, very high chance of dying from perpetual fever or what they call childbed fever. So he took it upon himself. He believed in microorganisms, although it had not been officially disproved or approved that, microorganisms exist. He believed that they existed. So he took it upon himself to simply start washing his hands between patients. And upon doing so, he drastically reduced women's death rates simply by washing his hands. So he tried to spread the word and get other physicians to wash their hands. And he was, again, met with a lot of criticism. People called him crazy. And so other physicians chose not to wash their hands because they didn't see the need to. And so there were still a lot of deaths. However, because he started washing his hands between patients, death rates dramatically started decreasing with his patients. He actually wrote a book about it, and he published a lot of this in his book. However, his book was also faced with a lot of criticism. You can go look him up, and there's going to be a short video that I have you watch on him, but it did not end well for him. Now, moving on to Joseph Lister. He had read some of Pasteur's work at this point in time. He had also read some of Semmelweis's work. He did believe that there was something in the air that was causing sickness. So not only did he wash his hands, but he was one of the very first cases where a physician, or a surgeon in this case, would disinfect their instruments between patients. Because again, oftentimes instruments would be used by numerous patients. They were never sterilized, they were never cleaned because the doctors, the physicians, the surgeons, they didn't know that they needed to be cleaned. So he started dipping his instruments in something called carbolic acid, which we know today, it's also known as phenol. And simply by doing this along with washing his hands, he drastically reduced death rates. So the process of simple hand washing along with dipping instruments in carbolic acid allowed people to start actually surviving some of these surgeries that usually had about a 90% death rate. So this was a little bit about some of the early cases or a little bit of early history of medical microbiology. Here's that video link that I was talking about. You might be able to copy and paste it. I know you can't directly click on it. You might be able to copy and paste it. I will try to send out an email with this video link on it because it's a 10-minute video. Just watch the one on the top. It's a 10-minute video, but it talks about Semmelweis and Lister, and it's very, very informative. It gives a lot of really good information. I really highly suggest that you watch this video. All right, moving on to Robert Koch. Now, Robert Koch, he was a scientist. He was working with bacteria. He worked with the deadly bacteria Bacillus anthracis, which causes anthrax. We still have cases every now and then of anthrax floating around. But he worked with bacteria to prove the germ theory of disease. The germ theory of disease is really simple. All it states is that if somebody got sick, there had to have been contact with some type of germ that made them sick. That's all it states. Because back in these times, it was generally thought that if a person got sick, that this was God's punishing them for being bad. That they had lied or somehow sinned or they cheated or they stole. And so God's punishment was to make you sick. Okay. Robert Koch came along and he said, no, this is not God's punishing you for you doing bad. You got sick because you came in contact with some type of pathogen that actually made you sick. And so for all of his work, he actually won the Nobel Prize in 1905. Now, he also came up with something called Koch's postulates, which we'll talk more about in a later chapter. A postulate is basically a fancy rule, okay? And these rules have to be followed in order. There are 4 Koch's postulates, and we'll learn those postulates later on, but it's basically an organizational or systematic approach to proving that microorganisms cause disease. And he showed that anthrax was caused by the bacteria Bacillus anthracis in 1875, again helping prove the germ theory of disease. All right, let's switch gears just a little bit. We're still talking about medical microbiology, but now let's talk about the birth of vaccination and the earliest understanding of the immune system. So there was a physician by the name of Edward Jenner, and he would oftentimes make house calls. There was also a disease at that time known as smallpox, which is caused by a virus. And the thing about smallpox is it's very, very deadly. It has a very high death rate, pushing 90%. Smallpox would come in and it was making like ghost towns because it was killing so many people. We didn't have any kind of medicine yet, so there was no cure for it. There was no preventative measure for it. But what Edward Jenner noticed was he was in an area that had a lot of dairy farms. And on these dairy farms were milkmaids. A milkmaid was a female whose sole job was to milk cows. All day long, all they did was milk cows. He noticed that when smallpox would pretty much kill everybody in a town, the milkmaids were always unharmed. They never contracted smallpox and died. And he was like, well, I wonder why. So what he did was he did notice something, that there was a disease somewhat like smallpox known as cowpox. And cowpox would infect the cows. It would cause these pustules to form on cows. You could pop these pustules. They would ooze pus. The cow would get sick, but they never really died of cowpox. He also noticed that when a milkmaid had been exposed to the cowpox, they would get very slightly infected, but they would always recover because cowpox doesn't really infect humans to a great extent. So he thought that there was somehow a correlation between smallpox and cowpox, because since these milkmaids had been exposed to cowpox, they somehow was not contracting smallpox. So he wanted to test his theory. So what he did was he went and got the gardener's son. The story goes the little boy was 8 years old, and he was used as a test subject to test Edward Jenner's hypothesis. So what Edward Jenner did was he went to a cow that was currently infected with cowpox. He busted the pustule. He got some of the pus. He went to this 8-year-old little boy. He scratched the inside of the arm of this little boy, and he put cowpox inside the arm, the open wound. Well, the little boy, he didn't really get sick. He was completely fine because, again, we don't really contract cowpox. The area got red, but it healed just fine. The little boy didn't get sick. Well, here comes the really unethical part. He goes to a patient of his that has smallpox. He punctures a pustule. He gets some of that very deadly smallpox pus. He goes back to the 8-year-old little boy, scratches the inside of his arm, and inoculates the inside of his arm with, again, this very, very deadly smallpox. Well, if Edward Jenner's theory is correct, the little boy should not contract smallpox because he was exposed to cowpox. Luckily, Edward Jenner's hypothesis was correct. The little boy got sick, but it was a very mild case of smallpox. He recovered and he was just fine. Ethics, however, says that we don't take a very deadly disease and inoculate a healthy kid just to see what happens. But because he was correct, Edward Jenner started going around the people in his village that were still living. He would scratch their arm and he would put cowpox or pus from a cowpox inside the villager's arm, and so the villager then could not contract smallpox, and it was very, very effective. They still at this point did not really know how the immune system worked, but he had an inkling that there was some protection offered by the human body from from smallpox, because the human body had been exposed to cowpox. It wasn't until about 100 years later, once again, we have the name Louis Pasteur shows up, because Louis Pasteur comes and shows how vaccines work, and he created the first true vaccine with a true understanding of the immune system and how the vaccination process works. So we give Edward Jenner the credit for the idea of understanding kind of the basics of immunity, but we give Louis Pasteur the credit for creating the first true vaccine. Okay, moving on to the birth of modern chemotherapy. All right, so first of all, let's talk about that term chemotherapy, because it means something different in microbiology. Normally, when you think of the term chemotherapy, you think of anti-cancer drug. Well, that's not necessarily the case in micro. Chemotherapy is designed, it means any drug that is designed to treat an illness. So we will talk about chemotherapeutic agents later on. So again, this is any medicine that's designed to kill some type of microorganism. So your antibiotics are chemotherapeutic agents. Okay. All right. So let's talk about the first drug. The year is 1910. There was a scientist by the name of Paul Ehrlich who developed one of the very first true drugs. It was a synthetic arsenic-based drug known as salvarsan, and salvarsan was used to treat syphilis. Okay, a little word about syphilis. Okay, so as I was saying about syphilis, syphilis is a sexually transmitted infection. And we've had major epidemics of syphilis in the past. During the Civil War, which happened in the 1850s, up through the Vietnam War, World War I, World War II, we've been having major outbreaks of syphilis. And a lot of our soldiers have had it in the past. And then also a lot of our big cities, such as New York City, Atlanta, places in Los Angeles, New Orleans, they would have major outbreaks of syphilis. The thing about syphilis is that syphilis occurs in 4 stages. The first stage of syphilis, you generally don't know you have it. The second stage of syphilis, you might have mild symptoms, but at this point they're still mild enough that you wouldn't automatically think syphilis until you reach the third stage, where you go blind. And then the fourth stage causes death. So syphilis, a lot of people were going blind right around the turn of the century, right around 1900. A lot of people were going blind from syphilis. And So Paul Ehrlich developed this drug, salvarsan, and it was designed to treat syphilis. And if you see in the upper right-hand corner here, this is a picture of syphilis and what it looks like in the microscope or under the microscope. Syphilis is long and thin, and it has like this wavy appearance. So syphilis is a bacteria, and so, again, we did not have any antibiotics at this point in time. That doesn't come till later on. So we did have this drug salvarsan, which was the first drug that we currently knew of that treated some type of microorganism. But the problem is with salvarsan itself, it was an arsenic-based drug. And arsenic, as we know now, is very, very toxic. It can be deadly. And so there were some people that were actually, they would take the salvarsan and then they would wind up dying from the arsenic that was in the salvarsan. But overall, salvarsan did do more good than bad, and so salvarsan was used quite a bit on the market. Now, salvarsan is not used anymore. We have better methods, we have better medications that are used to treat syphilis. What's very interesting about syphilis is syphilis was on the decline. After we had the invention of antibiotics, after salvarsan, syphilis rates started to decline, and it had declined to maybe 1%, less than 1%, basically to where it was non-existent. However, something interesting happened about 7 years ago. For the first time in 70 years, syphilis is now on the rise again. Okay, so syphilis is now yet another disease that we are on the lookout for. Alexander Fleming, the year was 1928, he accidentally discovered the first antibiotic. And like I mentioned earlier in this chapter, the first antibiotic was penicillin. Okay, and it was an accidental discovery. Here's a picture of Alexander Fleming over here on the right. And you can see in the background, he has all of these Petri plates. And it looks like he has something growing in those Petri plates. And we'll talk on the next slide, we're going to talk about Alexander Fleming and his accidental discovery. So, and then after 1928, in the 1930s, we had, for the first time, we had the synthesis of the first sulfonamides. These are the sulfur-based drugs. And sulfur-based drugs are still used today. We have made drastic improvements on the sulfonamides. But if you've ever been to the doctor and they asked you if you were allergic to sulfur, it's because they were about to prescribe you a sulfonamide. So again, they are very effective and they're still widely used. But let's go back a little bit and let's talk about Alexander Fleming and his accidental experiment. So Alexander Fleming was a microbiologist and he was working with the common bacteria Staphylococcus aureus. He had taken staph. I typically will just abbreviate it staph. And he had inoculated a stack of Petri plates with staph. And then he waited a couple days. He waited for the staph to grow. And then he went back to that stack of Petri plates and he was looking at each one of the Petri plates to make sure that the staph grew when he came across a plate that looked something like this. This would have been the staph. Each circle, those are called colonies. So this Petri plate had colonies of staph, which is what it should have had. This is what he was trying to grow, was the staph. But he noticed on one of his plates that some kind of contamination had landed and had grown on one of his plates. This was the contamination. He didn't, at that time, he didn't know what it was. Okay. He just knew something from the environment had landed on the plate, contaminated the plate, and it started to grow. But he was about to throw the plate away, thinking it was no good because it was contaminated. But he noticed something. He noticed that the way he had the plate set up, the staph should have grown over the entire surface of the Petri plate. But what he noticed was where the contaminant was, there was this zone here where there wasn't much growth. That means he was thinking that this contaminant, whatever it was, had somehow suppressed the growth of the staph because there was no growth in that area. which led him to further study the contaminant. Well, he named it. It was discovered that that contaminant was a fungus known as Penicillium nonatum. And it took a while because this accidental discovery occurred in 1928. But it wasn't until 1945 that penicillin was actually put in capsule form and released to the public. And it just so happens that our World War II soldiers were almost like guinea pigs. They were some of the very first people to ever receive penicillin. So luckily, penicillin was a good drug. The soldiers reacted very well to penicillin, and then penicillin was released to the masses based on results from our soldiers. So that is how penicillin was accidentally discovered by Alexander Fleming. Okay, modern developments in microbiology, so current times. We're going to start off at the top with just some terms. Bacteriology is the study of bacteria. Mycology, this is the study of fungi. Parasitology is the study of parasites. Virology is the study of viruses. And immunology is the study of the immune system and how the immune system mounts a response to invading microorganisms. So one of the first, what we call modern developments, is genetic engineering or recombinant DNA technology. Recombinant, it sounds like recombining because that's what they're doing. They're taking DNA from two different organisms and they're recombining them or combining them together. And this is genetic engineering. The first time genetic engineering took place was in the 1960s. What they did was they took DNA from one bacteria species and DNA from a second bacteria species, and they tried to combine the DNA from the two different species. There was no real point to this other than just to see if it could be done or not, because it had never been done before. And it was successful. The experiment was successful. They had successfully spliced together or recombined two different species of bacterial DNA. So then, this was 1969, I believe. And then in the 1970s, 1971, they attempted to redo this experiment. But instead of doing it with prokaryotes, bacteria, they did it with very simple eukaryotes. Again, they took eukaryotic DNA from one organism and eukaryotic DNA from a second organism, and they attempted to combine it together. Again, just to see if it could be done. These were the very earliest stages of genetic engineering. From that, it's pretty much taken off, and we have performed a lot of genetic engineering to date. A lot of our food that we currently buy has been genetically engineered. Our corn, our crops have been genetically engineered, usually for two reasons. Genetic engineering helps the crops grow larger. So your ear of corn, if it's been genetically engineered, it will grow larger than it would normally grow, and that means more money for the farmer. Also, a lot of our crops have been genetically engineered to be resistant to pests or be resistant to certain diseases because there are fungal diseases that affect our crops. And so what if you were to able to make that ear of corn resistant to diseases? Well, again, that means more money for the farmer. So that is why a lot of our food has been genetically modified. I gave you the example earlier of E. coli and how insulin is now actually produced through genetically engineered E. coli cells. They took the gene that codes for insulin production, they inserted the gene into E. coli cells, and so now the gene is expressed in these E. coli cells. So the E. coli cells are now producing insulin, and then the insulin is collected, it's purified, and then it's distributed to pharmacies for people's use. So that's just a couple of examples of genetic engineering. You hear new examples all the time. Okay, so on this slide right here, you do not have to memorize anything, okay? I just put this up here so you could see some of the years, the scientists, and the Nobel Prizes in the area of microbiology. So we have there's been a lot of research, a lot of knowledge that we have obtained in the area of microbiology since the turn of the century. One of the latest was in 2008 with HPV and cancer. This is where human papillomavirus was proven to cause cervical cancer in women. Okay, and so that was some of the latest in micro research where a Nobel Prize has been won. Okay, so the term microbiota in red. Now next to that you see the term flora in parentheses. Flora is an old-fashioned term. Up until the 1930s and 1940s, bacteria were referred to as flora. Okay? A more modern term is microbiota. This is simply all the bacteria, all the fungi that are naturally occurring in a person's body. There's an estimate that says the average person contains 2.2 pounds of bacteria. Okay? There's several hundred species of bacteria that live in the mouth alone. We have species of bacteria that live on our skin, that live in our nose, that live in our throat, that live in our intestines. The only place in the human body that is not supposed to have bacteria is the blood. If you have bacteria in the blood, you have a major, major problem. Okay. But other than that, there's pretty much bacteria throughout the entire body. This is considered normal bacteria because it's living on you or in you and it's not causing any problems. Now, every now and then we come across pathogenic bacteria. This is where there are certain pathogenic strains of bacteria that enter the body and they're able to overcome our immune system. And then they lead to infectious disease. Now, antimicrobial resistance. This is a topic. This is a big topic that I'll be talking about quite a bit throughout the semester because we hear about antimicrobial resistance all the time in the news. We hear about things like MRSA, which we'll talk more about in a minute. We are hearing about diseases where once we had a cure for particular diseases, now there is no longer a cure because antibiotics have been overused so much. So again, a very big topic right now. Another topic is bioterrorism. It's been a few years ago, but there was a time when anthrax was being mailed in the mail. I remember they were saying on the news, you know, if you receive a package and you're not expecting the package, don't open it because some of these packages were containing a white powder. In this white powder was anthrax, and people were inhaling the anthrax and they were coming down with anthrax. And anthrax is a very deadly disease. So there's been other ideas or other potential bioterrorism opportunities, another one being with smallpox. So we talked about smallpox and Edward Jenner. Well, a vaccine was created for smallpox and the world was eradicated from smallpox. So there are no more active cases of smallpox. But there's two places where that still contain a sample of smallpox. One is in Atlanta, Georgia at the CDC. The other is in Moscow, Russia. And there's been talk about what would happen if one of these strains of smallpox was somehow released into the environment, because we are no longer vaccinated for smallpox. Okay, re-emerging diseases, things like the Zika virus, WNE, which is West Nile encephalitis, avian flu, MRSA, BSE, which we'll talk more about, E. coli, HIV, AIDS, and now the big one, COVID-19. So just a word on COVID. I did not make a slide specifically for COVID, but just a word on COVID. COVID-19 is from the family coronaviruses. The coronaviruses were first discovered in the 1960s, so the coronaviruses are not new. We've known about them for a while. COVID-19 is a new strain of the original coronaviruses. We still don't know the exact origins of it. There's a lot of theories out there. But just so you know, coronaviruses have been out for a while. COVID-19 is just a new strain. And yes, it's wreaking havoc worldwide. We've had high fatality rates, particularly in hot spots like New York City. New Orleans was a hot spot. Atlanta, Georgia is considered a hot spot. So that's a new disease because it is considered to be a new mutation. But let's talk about a few of these diseases in a little more depth. So West Nile encephalitis, it was caused by the West Nile virus, okay? It's not new. It's been around since 1939. That was when we discovered the first case of it. However, it was pretty much only located in Africa. Well, due to the ease of travel, people are able to travel cheaper and they're able to go more places. So it was thought that somebody had traveled to Africa, and when they landed in New York City, they were infected with West Nile encephalitis. And so it spread to other people. Encephalitis means inflammation of the brain and spinal cord, which can be very, very deadly if not treated very quickly. But that was pretty much an isolated case or an isolated outbreak of it because, again, West Nile encephalitis really only occurs in Africa. It was brought back to the United States by a human host. Avian flu. This is another big one because the avian flu also originated in China. The year was around 2000, between 2000 and 2005. Avian flu. Avian means bird. This is the bird flu. Now, the bird flu, again, is nothing new. Bird flu has been around for a very long time, but it only infected birds. Well, for the first time in history, a bird virus jumped to a human. A human became infected with the bird flu. There were a lot of questions asked. How did the person contract a bird virus? Is it deadly? Can the person spread it to another person? What are the symptoms? So do we have treatment? Is the person going to be okay? I mean, there were so many questions because we have never seen a bird virus in a human before, or this particular virus anyway. So we had a lot of unknowns. So a lot of time in the first few cases, they were human guinea pigs, basically. We were watching them because we didn't know anything. We didn't know how they contracted it. It turns out that we will learn later on that viruses mutate at a very quick rate. And viruses are constantly mutating. And they think that the virus mutated to a point that it could jump to a new species or a new host. So it jumped from a bird into a human for the first time because the virus mutated. And when I say mutated, what I mean is that there was some type of change in the DNA. There was some mutation made, that DNA, it got changed. Remember, there's 4 nucleotides in DNA. You have adenine, thymine, guanine, and cytosine. And something happened along the way, and there was some type of change made in that DNA, which now allowed it to jump to a new host, and that new host just happened to be a human. It also came to the United States. It also landed in New York. You will learn that New York is a hot spot for new viruses, and the reason for this is because New York is very, very crowded. There's 7 million people that live in Manhattan, which is like a 13-mile space. And then not only that, but New York is a hot spot. It's a hub for international travel. And so a lot of international flights will land in New York, and then you transfer to your domestic flight. So a lot of diseases will make their first appearance in New York. But they have not found human-to-human transmission has not been found with the bird flu. So they think it was a particular strain of the bird flu that had mutated to a form that could jump to a human. I've heard about new cases of the bird flu in birds, but I haven't heard about any new cases of the bird flu appearing in humans. MRSA. Okay, MRSA stands for methicillin-resistant Staphylococcus aureus. So it's a particular type of Staph aureus. So when we had our first antibiotic, penicillin was used. It was the only antibiotic that we had. So it was given out for any type of disease because we didn't know it didn't treat viruses at that point. So it was pretty much given out. If anybody had any type of illness, they were given penicillin. Well, the problem was, remember, penicillin was only released to the masses around 1945. And then in 1950, we had already started seeing penicillin resistance develop because it had been overused so much. It had been given for everything. And then, so in the 1980s, between the 1950s and the 1980s, there were several new antibiotics that came along. But one of those antibiotics was known as methicillin. Methicillin is a derivative of the original penicillin. And methicillin was given, again, to kill Staph aureus. But methicillin resistance occurred very quickly. So in the 1980s, we started seeing methicillin resistance to Staphylococcus aureus. And as a side note, methicillin is no longer even used in the United States. It's used in certain other countries. It's used in England, for example, but it's not used here in the United States. And then in the 1990s, they started seeing something known as MRSA, which was not only resistant to methicillin and penicillin, but is also resistant to an antibiotic known as vancomycin. Up until recently, vancomycin was considered to be the strongest antibiotic that we had. It's so strong that it can also shut down your liver, and there's a lot of bad side effects that go along with it. So they tried to limit the use of vancomycin because vancomycin has toxic side effects, but also we'd already started seeing resistance to it, and it's really the only other drug we had on the market that would kill the superbugs, what we're now calling superbugs. So Staphylococcus aureus, we started seeing vancomycin resistance to MRSA in the 1990s. Since the 1990s, 2000s, 2010, now we are seeing strains of MRSA that are resistant to everything, that simply cannot be killed. There's roughly 30 different antibiotics that we use here in the United States, and there are strains of MRSA going around that are resistant to all 30 antibiotics. They're resistant to combinations of antibiotics. And so we'll be talking more about MRSA and antibiotic resistance because, like I said, that's a very big topic. Next, bovine spongiform encephalopathy. This is the scientific name for mad cow disease. Okay. It was named spongiform encephalopathy, spongiform because it causes spongy plaques on the brain. Encephalopathy meaning basically brain, so it causes spongy plaques to form on brain tissue. This is a disease that is caused by a prion. Now remember, I said earlier that prions are nothing more than an infectious protein, and they have a 100% death rate. Now, there's only a handful of diseases, maybe 5 diseases that are caused by prions, and we'll cover all of those prion diseases later. But this is just the one of the newer ones because prions weren't discovered until the 1980s, and then we had the mad cow outbreak in 1988 over in England. There is a human version of the mad cow disease. It's pronounced Creutzfeldt-Jakob, so it's pronounced differently than it looks. Creutzfeldt-Jakob. This is the human form of mad cow disease. So again, it's very, very rare, but it is prion-based, and it does have a 100% fatality rate. Okay, moving on to Escherichia coli 0157H7. So to start off, I will say that there's over 200 strains of E. coli. A lot of them are harmless. We have E. coli living in our intestines right now, and we're not sick. So a lot of the strains of E. coli are perfectly harmless. They're considered to be normal microbiota. However, in 1982, they discovered a particular strain of E. coli, and they named it 0157H7. This is considered to be the deadliest or the most toxic strain of E. coli that currently exists. It causes diarrhea, and the diarrhea gets so bad that the person usually dies of dehydration. It really tends to affect the children as well as elderly. We don't see it too often in the United States because E. coli is said to be a fecal bacteria. So it would require us coming in contact, us consuming this fecal bacteria. And we wash our hands. We have decent health practices over here in the United States compared to other places. And so E. coli in general, particularly the pathogenic strains though, they tend to be far more common in other countries. One thing about E. coli that a lot of people don't realize is every now and then you do hear about a food being recalled, and it's usually due to either E. coli, Salmonella, or Listeria. Those are the three common bacteria that are responsible for food being recalled. And it's because with E. coli produces a toxin, and you can see that toxin here in purple. So that's the toxin that E. coli produces. So when a person gets sick from E. coli, it's usually not so much the bacteria that they're getting sick from. They're getting sick from the toxin that the bacteria produces. And so, for example, if you're cooking, if you're grilling hamburgers or something like that, yes, the heat, the heat will kill the bacteria. But in order to break down the toxin that the bacteria produces, you have to boil it for 30 minutes. Most people aren't going to cook a hamburger at boiling temperatures for 30 minutes. And so that's the big risk with E. coli, is it's not so much the bacteria itself, it's the toxin that the bacteria produces that causes the actual sickness and the diarrhea. Okay, last slide. Let's talk a little bit about HIV/AIDS. So it's caused by a virus known as human immunodeficiency virus. It was first identified in 1981 in San Diego, California. There were 5 people that showed up with some mysterious illness. They had no idea what it was. These 5 people died. Blood samples from these 5 people were stored so that later on, when we had more knowledge, or if we came across something similar, we could pull out these blood samples and compare it to current cases. It is a worldwide epidemic infecting 30 million people, 14,000 new infections every day, and right now the country where HIV/AIDS is growing the fastest is once again China. So China is facing a major HIV epidemic right now. It is considered to be sexually transmitted, infecting both males and females. If you look at this picture over here, I wanted to point out, now this data was from 2011, but I found newer data from 2016, and the data is still the same. There are 5 states that make up almost half of the new cases of HIV. We have New York, Florida, California, Texas, and Georgia. So yeah, Georgia is on this list. We, along with the other 4 states, make up almost half of the new HIV cases, and that data is still the same even if you look into 2016. Georgia, we rank 5th, but still 5 out of 50 is not that great as far as new cases. Atlanta, again, tends to be the hot spot of new HIV cases. And I think that is all I have to say about Chapter 1. Okay. Again, if you're reviewing this, if you're listening to this and you have any questions, feel free to email me, feel free to message me on Blackboard. So far, we've been talking a lot about different bacteria. We've talked a little bit about the history of bacteria. We've talked about things that we'll be studying this semester. Well, let's just talk a little bit about bacteria, not only bacteria, but fungi, the protozoans, and talk about how these microorganisms shape our planet. We've already talked about how some microorganisms are able to perform photosynthesis that releases oxygen that we are able to use, but they also produce carbon dioxide, nitrous gas, different types of methane, different types of alcohols come from or byproducts of bacteria. So you can literally find bacteria and other microbes everywhere on planet Earth, except for in the blood. The blood is one place that is supposed to be sterile at all points in time. If microorganisms make their way to the bloodstream, that is a condition called sepsis, which we'll talk about in a later chapter, which is a very serious condition. But other than that, as far as bacteria and other microbes such as fungi, they live on our skin, they live in our intestinal tract, they live in our nose, in our mouth, in our ears, in our eyes. They're everywhere, literally everywhere, except for in our blood. There was one fact I came across that said the average person contains 2.2 pounds of bacteria. Bacteria are said to be the most abundant cellular organism, but remember we talked about viruses. Viruses are acellular, but it is thought that there's actually more viruses here than any type of cell. All right, the last section of chapter 1 deals with the macromolecules, the 4 main groups of the macromolecules. Remember, macro means large, so these are going to be large molecules. Okay? There are 4 main groups. There are the carbohydrates. This includes things like your sugars. And then we'll talk about lipids, which are your fats and your cholesterols. We will talk about proteins and their 4 structures, or 4 different levels, I should say. And then lastly, we'll close with talking about nucleic acids, such as your DNA and your RNA. So first of all, let's talk about a monomer versus a polymer. A monomer is a small, simple subunit. You can have many monomers come together to form a polymer. So an example, if we're talking about proteins, I've said earlier that a protein is nothing more than a chain of amino acids. So you can have a whole bunch of amino acids. These would be the monomers. And when you take these amino acids and combine them together into something larger, that now becomes a polymer. So a polymer, poly meaning many, a polymer is made up of many monomers. All right, as far as carbohydrates are concerned, there are a lot of different carbohydrates. We have our sugars, glucose, fructose, sucrose, dextrose, mannose, lactose, just to name a few. Okay, and then we have our complex carbohydrates such as our starch, our cellulose, and things of that nature. So carbohydrates can be broken down into something called monosaccharides. Mono, again, meaning one. These are our simplest sugars. Glucose would be a good example of a monosaccharide. Okay? But then we also have our disaccharides, di meaning 2. This is where we take 2 monosaccharides and we stick them together with a bond. Okay? Sucrose is a really good example of a disaccharide. You can also have polysaccharides, poly meaning many. This is where you have many sugar units that come together. Again, I mentioned starch and cellulose, but glycogen is another one. Most carbohydrates go by a particular formula. You can see CH2O, and then the entire thing is in a parentheses, and then there's a sub N. Okay, so that's the general molecular formula for any type of carbohydrate. Most sugars end in ose. So hexose, well, the prefix hex means 6. So hexose are 6 carbon sugars. Pentose, well, pent means 5. So pentose are 5 carbon sugars. Then we have glucose. We will talk a lot about glucose when we talk about aerobic respiration, because glucose tends to be the main sugar that the body uses as well as bacteria. So if any sugar can be broken down to glucose or converted into glucose, it's done so. But then we also have fructose. This is why our fruit is sweet. We have xylose, which is a wood-based sugar. Lactose, which is milk sugar. Maltose, which is malt sugar. And then we have sucrose, which is table sugar. So these are all different types of sugars. And then over on the right, you see the picture, and you can see the difference between a monosaccharide, a disaccharide, and a polysaccharide. So a little bit more on polysaccharides. Remember, they're just long chains of carbohydrates, usually glucose. So there are several that are important in microbiology that are considered to be polysaccharides. Cellulose, okay, we've talked about cellulose. It's composed of plant cell walls. It's also found in some algae. And then agar is a very important ingredient in culture media as far as microbiology is concerned. This is what we grow bacteria on, is agar. It's nothing more than a complex carbohydrate or a polysaccharide. Chitin, we've talked about chitin because we talked about fungi and how fungi have cell walls made of chitin. Well, chitin is another polysaccharide, as is peptidoglycan. There's that long word again. I said the only place where you find peptidoglycan is bacteria. Bacterial cell walls are composed of peptidoglycan, which is nothing more than a polysaccharide. Lipopolysaccharides. Near the end of the semester, we are going to talk about toxins. And there are certain types of toxins that are only found on a specific type of bacteria called the gram-negative bacteria. Well, these gram-negative bacteria are unique because they have an outer cell membrane that contains the lipopolysaccharide. So don't worry about it too much yet. We're definitely going to come back to it though. And then the glycocalyx. Starting in the next chapter, we talk a lot about the glycocalyx. Again, it's a very important structure that some bacteria have. It's found on the outside of the bacteria cell. It's sticky, it's sugary, and the bacteria cell is able to use their glycocalyx to bind hold to other surfaces and other structures where they may be located. So it also works to protect the bacteria cell. So it's an outer layer, which is called the glycocalyx. Again, it's nothing more than a complex polysaccharide. Lipids. These are your fats. Now, I've been talking about lipids. I talked about the term organic. Organic means that it contains carbon with a bunch of hydrogens around it. Well, here, if you look at the structures of your lipids and your fatty acids, you see there's carbons and they have hydrogens around them. So your lipids definitely fit the definition of being organic. Okay. Now, fats can be saturated or they can be unsaturated. A saturated fat means that all it has are single bonds. If you look at the picture on the bottom, the picture on the left in the bottom picture, you will see there's a straight chain of nothing but carbons and hydrogens. That is said to be a saturated fatty acid because there's nothing there but single bonds. However, if you go to the picture over here on the right, let me see if I can mark it. Right here where I'm marking, there's a double bond there. That double bond throws off the whole structure. It's now got a bend or a crook in it. That is said to be unsaturated fats. So that's the difference between saturated fats and unsaturated fats, is you're looking for the presence of double bonds. If there's double bonds, then it's said to be unsaturated. Okay. Phospholipids. This is another lipid, aka fat. The phospholipids become very important when we talk about the cell membrane of bacteria. You learned in anatomy and physiology that human cells have a cell membrane composed of a phospholipid bilayer. Well, bacteria also have a phospholipid bilayer. And you can see it's very much like this structure in this picture here. You have hydrophilic regions. Hydro means water, philic means loving. So water-loving regions. Those are going to be the head regions, which are the orange ball or sphere-shaped structures. And then you have your hydrophobic regions. Hydrophobic means water-fearing. These are going to be those long tails of fatty acids. It doesn't matter whether it's an unsaturated fat or a saturated fat. They do not like water. Okay. So it's always going to orient themselves so that you have a phospholipid bilayer. Bi meaning two, because you'll have two single layers of these phospholipids and they arrange themselves so that those hydrophobic tails face each other, creating a hydrophobic compartment on the inside. So we'll definitely come back and we'll talk more about these phospholipids. I believe it is in chapter 3. Just a very brief word on steroids and waxes. Steroids, a good example of a steroid is cholesterol. Okay. Steroids are found in various places inside the bacteria. It gives stability to the bacteria. It gives shape to the bacteria. It also helps reinforce cell membranes in bacteria. Waxes, okay, also a form of lipid or fat. They involve long alcohol chains and saturated fatty acids. These waxes are very good because they act in waterproofing. So anything that's waterproofing, whether you've got examples like fur, feathers, fruits, leaves, human skin, insect exoskeletons, shellfish exoskeletons, the wax prevents the uptake or the absorption of water. So it acts in waterproofing. Okay. We will talk about a very special class of bacteria that has a unique layer of wax that covers the bacteria. They are called acid-fast bacteria, and they will be introduced in the next chapter. It just so happens the bacteria that causes tuberculosis is said to be acid-fast. It's very unique because it has a waxy layer in the surrounding known as mycolic acid. So all of these are definitely topics that we will come back to more later on as the semester progresses. Okay, another big category are proteins. All right, so these are the prominent or predominant organic molecules in cells. They are all composed of 20 amino acids. Now, there has been some studies that have stated that there's 21 amino acids or even 22 amino acids, but most classic literature states that there's only 20 amino acids. Your book says that, so we're going to go with 20 amino acids. So the difference between proteins, peptides, polypeptides, it's simply the number of amino acids. So if you have just a couple amino acids stuck together, that is known as a peptide or a peptide chain. Now, polypeptide, the prefix poly we know means many. So if you have 20 amino acids and they are stuck together, it is called a polypeptide or a polypeptide chain. Proteins usually have about a minimum of 50 amino acids. However, some proteins can get extremely large. They can have as many as 2,000 different amino acids. So basically these are amino acids, but there's a large quantity of amino acids and they're held in a particular shape to give that protein structure and therefore function. Let's talk about the different levels of protein structure. Because with proteins, you have a primary level, you have secondary level, you have tertiary level, and you have quaternary levels. So let's talk about the first two on this slide. So your primary chain is simply the chain of amino acids. Okay, that's all it is. There's no bonding that takes place. It is a straight, long chain. You can think of pearls on a necklace, each pearl being an amino acid. Okay. Next is your secondary structures. The two most common secondary structures are your beta-pleated sheets, which I'm marking, and your alpha helixes, which form a helical type structure. They are held together by very simple bonds called hydrogen bonds. Hydrogen bonds break very easy. You can break them very easily with heat or acid. So these secondary structures are only held together by hydrogen bonds. And again, the two most common types of secondary structures are your alpha helixes and your beta-pleated sheets. All right, if we move on and we talk about tertiary structures and quaternary structures, tertiary structures are fully functional proteins. You can see one on the left-hand side here. Um, and the protein is bound like it should be bound, and it is fully functional and fully operational. There are a lot of different types of bonds that hold a protein together in its tertiary state. So yes, there are hydrogen bonds, but we also have ionic bonds, we have covalent bonds, we have van der Waals forces, we have disulfide bridges. So all of these different types of bonds come together to fold the primary structure of the amino acid into the tertiary structure of this protein so that this protein is functional and active. Lastly is our quaternary structure. Quaternary structure, quant, the prefix quant means 4. This is where we have more than one chain of polypeptides, but they come together to form a really large functional unit. So you can have 3 different strands or even 4 different strands of polypeptides that combine together, and they're all held together by a variety of different bond types to give the protein its structure and therefore its function. So like I was saying, proteins are held together by a variety of different types of bonds. When a protein is said to be in its native state, that means that protein is fully functioning, it's working correctly, all of the bonds are being held together that give that protein its shape and its structure and its function. However, when you break those bonds, causing that active protein to unravel, that protein is now said to be denatured. A denatured protein does not work. Okay? The amino acid sequence is still correct, but because all of the bonds are broken, the main shape of the protein is not functional anymore, and therefore it's not active, and therefore it does not work. There are several ways in which you can denature a protein. You can add heat to a protein because we know that heat breaks bonds. Okay? Acid, another very common one, acid breaks bonds. You can use alcohol and even some disinfectants, and that's how they actually work to kill bacteria when you're trying to disinfect a surface, is a lot of these have action on proteins or maybe the cell membrane of the bacteria cell, and they work by denaturing proteins. But again, probably the easiest way to denature a protein, the most effective, fastest way, is with heat, because you're immediately breaking all of those bonds that hold that protein together. Okay, the last section of this chapter deals with nucleic acids, namely DNA and RNA. We'll basically talk about DNA here, and then when we cover the genetics chapter, we'll talk more about DNA and the RNA. But DNA is basically the coding center of a cell. It gives all instruction on the activity of that cell. It states when a protein needs to be made. It states when an enzyme needs to be made. It tells that the rest of the cell what to do. So if you look at the backbone of DNA, it's composed of alternating sugar phosphates. So you'll have a sugar, and then you'll have a phosphate, and then you'll have a sugar, and you'll have a phosphate, and that forms like what they call a ladder, or the backbone of the ladder. And then you have these nitrogenous bases. There's 4 of them. We have adenine and thymine, guanine and cytosine. So again, these are called nitrogenous bases, and they bind in a particular way. Adenine always binds with thymine. Therefore, thymine always binds with adenine. And guanine always binds with cytosine. And they bind together with hydrogen bonds. Okay? And so this is how DNA is formed. You have the alternating sugar phosphate backbones, and then you have these nitrogenous bases that form like the rungs of the ladder because they pair together in such a way that it binds and forms a really nice helical ladder-like structure. You can see that helical ladder-like structure. So if you were to unwind it, it would look like a really nice ladder. But again, due to bonds, DNA naturally has a helical nature to it. It looks like it curves or it curls. But here, if you look at this picture, this is what I was showing you or talking about on the previous slide, that the backbone is composed of sugar groups and phosphate groups, and that they form the backbone, and they alternate sugar, phosphate, sugar, phosphate, sugar, phosphate. But then coming off of the sugars, we have our nitrogenous bases, either adenine, cytosine, guanine, or thymine. And then they bind accordingly together to form that nice, uniform, ladder-like structure. The last little section of this chapter deals with ATP, which is short for adenosine triphosphate. Anytime I'm talking about energy in this chapter, I'm talking about ATP. So if I ever mention energy, just automatically know I'm talking about ATP. When we cover aerobic respiration and I'm talking about energy, we're making energy, I'm talking about making ATP. ATP is energy. Okay. So ATP is very important. It's composed of three main subgroups. So there's an adenine, there's a ribose, which is a sugar, and you have three high energy phosphate groups. And you can see the picture of, you can see the adenine and you can see the ribose sugar and those three phosphate groups out to the left of the picture. When those phosphorus bonds break, energy is released, but not just a normal amount of energy. An excessive amount of energy is released when those phosphate bonds are broken. That is the energy that we use to do work. Or in the case of bacteria, that is the energy that the bacteria use in order to do work, whether it's to move or whether it needs to make something, make a component for itself. So that is ATP, and we will talk more about ATP when we talk about aerobic respiration, which is in a later chapter. All right, that is all of chapter 1. I know it's a long chapter. There's a lot of material here, but that's why I record these lectures so you have a chance to go back and listen to them at your own convenience. So as always, if you have any questions, feel free to email me at any point in time, and I will try to get back with you. All right, so that ends chapter 1.